Yeah, so this is a picture of my two children. On the left is my son, who turns 6 next week, and on the right is my daughter, who is 16 months old. So let's see here. Here's a picture of me and my father at a family reunion, and it turns out in 2011 I lost him to pancreatic cancer. And it turns out he had lost his mom to stomach cancer. So there's a fun fact. Muriel's in the room. As everyone knows, Muriel was my PhD advisor. When I was finishing my PhD on my way to go to the University of Illinois to hang out with Sean Mine and everyone else, she told me the first thing I should do is call up the department chair, tell him I'm going to delay my start date, and I should go do a postdoc in something wildly different. So I ended up doing a postdoc in neuroscience. And that was an experience of what I call feeling comfortable, feeling uncomfortable, because I had not taken a class in biology since AP biology in high school. And then here I was as a neuroscience postdoc. But I got through it, was able to have some fun doing brain science. So when the situation occurred with my dad and, this disease of the digestive system, his pancreatic cancer and his mom's stomach cancer, I told myself, in the spirit of feeling comfortable, feeling uncomfortable, I'm going to go and learn something I know nothing about again. So I started to play around and look into what goes on with the digestive system. And if you see someone who has, GI problems are interesting because there's a small set of symptoms: pain, nausea, bloating, constipation, and yet you can have a variety of different types of diagnoses, as you can see here. And it takes a long time for them to figure out the diagnosis in many occasions. And it gives rise to huge annual costs, it's sort of, as you can see here, about $150 billion annually. So when I first began to think about this problem, at first glance, it wasn't obvious how I could contribute, because if you think about the digestive system, you think the stomach, acids. I'm not a chemist. You think about squeezing to propel food through the stomach, peristalsis. I don't do mechanics. But then I learned that the digestive system actually has 100 million neurons inside of it. And also there are electrical connections between the digestive system and the brain via things like the vagus nerve and the spinal cord. So I began to get more intrigued how this electrical engineer / neuroscientist could contribute to this problem. So a little bit about the stomach, for instance, is when food first goes down your esophagus, the top part of the stomach that gets activated is so you can see here's the fundus, and the fundus has stretch receptors, so it can expand for food coming in. And those stretch receptors send downstream neurotransmitter so that for the contractions occur in the stomach, they can get ready to produce. It also elicits acid secretion and other things so the stomach can begin operating. So in a normal operating stomach, that food gets broken down with those acids and those contractions occur. And when it's sufficiently broken down, there's a sphincter, just like when you pee, that opens up and allows the food to go into the small intestines. Other people have a condition known as gastroparesis, or delayed gastric emptying, and food basically gets trapped in the stomach longer than it should. This is just a very high level example. So I mentioned that the brain and the stomach are connected. So more specifically, when you're in a very stressed out state, your pupils get big, you start to breathe faster, your heart beats faster. It turns out that inactivates the digestive system. And on the flip side, when you're in a calm state and parasympathetic, in medical school, they call that condition rest and digest. So your pupils get smaller, you breathe more calmly, your heart slows down, and that hits the gas pedal to the digestive system. So one of the things that's interesting is if someone comes in and they have delayed gastric emptying, there could be many reasons why you have delayed gastric emptying. Maybe it's like, suppose your car doesn't operate the way it's supposed to. Maybe there's a problem with the engine of the car. Maybe the contractions are not contracting the way they should. Maybe there's a problem with the connection between the brake pedal and the engine. Like there's excess sympathetic activation because you're always stressed out. Or there's a problem between the gas pedal and the engine. For instance, diabetics, half of diabetics have delayed gastric emptying, in no small part because diabetes can elicit a neuropathy, including neuropathy of the vagus nerve. So that means that the connection between the gas pedal and the engine is off. Or in the spirit of comedy, if people remember the movie Beverly Hills Cop, remember there was a point where they said, I'm not falling for the banana in the tailpipe. So if you put a banana in the tailpipe of the car, even if the engine is operating fine, the car is not going to drive properly. So the gas pedal and the brake pedal could be operating fine, so could the engine. But if that sphincter is not opening and closing as it should be, that can be another reason you have these problems. So at first glance, when you come in and you have delayed gastric emptying, it takes a long time to figure out what this cause is. So one of the things that we got attracted to as, electrical engineers is there are these interesting cells called the interstitial cells of Cajal. If anyone knows a little bit about neuroscience, this guy named Ramon Cajal won the Nobel Prize for these beautiful images. He named these cells, and these cells are throughout the lining of the GI tract. And what's interesting is that they integrate information from the 100 million neurons in our digestive system, and they pass that information to the smooth muscle cells to contract. So the way they work is that, like neurons, they have ion channels that open and close, and they have a membrane potential. And when it crosses a threshold, action potentials get generated. What's unique about them, though, and let me see if this pointer works, is that, so if you see when it crosses a threshold, an action potential generates. And if there's excitatory input, like I mentioned, the parasympathetic nervous system, rest and digest, that's mediated by a neurotransmitter called acetylcholine, which is excitatory. Then the membrane potential, gets larger. But on the flip side, it can be decreased if there's sympathetic input via stress, and that's via inhibitory neurotransmitter like norepinephrine and nitrous oxide. Now, the one other unique thing about this picture, notice, is that this membrane potential is always oscillating, even when action potentials are not generated. So this membrane potential is always oscillating. And what's cool about this is that this basically coordinates timing of when contractions will occur. Contractions are more likely to occur at the peak of the oscillation. Why is that interesting? Well, if you look spatially across the whole stomach, for instance, it turns out that that membrane potential, if you look spatially, is actually a propagating wave. And so that is sequencing the timing of when contractions can occur. And so that's why you see the contractions going boom, 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 propelling food from the stomach into the small intestines. And and that is how it gives rise, to instance, for a bolus of food to go down your GI tract. So one of the things that we got interested in from 30,000 feet is that I've done EEG work before. You know what EEG has done for the brain, what EKG has done for the heart. Could we do something analogous for the digestive system? This was a 30,000 feet idea. You always walk on the shoulder of giants and ask yourself, who's done this before? So this technique got popular in the 1980s. And the way that the doctor suggested you do it is you place 3 electrodes. One is a reference, one is a ground. So the middle one is actually recording data. And you put it at the midpoint between the belly button and the bottom of the rib cage. And those pacemaker cells that I mentioned before, it turns out they always oscillate in the stomach at about 0.05 hertz. So if you, basically filter the data, what you can look at, for instance, is build a time frequency plot. Look after a meal occurred, you should see more energy at 0.05 hertz because those stretch receptors got engaged. They send the downstream neurotransmitter. So this was the idea of the 1980s, but then a bunch of publications came out and basically said this measure, the electrogastrogram, has poor correlation with clinical gold standards, doubtful usefulness. So the punchline is that people tried to do this, but didn't get meaningful information. So a fun fact, a terminology we came up with in the spirit of comedy, people with gray hair know who this is. This is a comedian named Rodney Dangerfield, who in the 1970s and 1980s would go on Johnny Carson and say, let me tell you why I get no respect. My wife gives me no respect. My kids give me no respect. So this electrophysiologic signal is the Rodney Dangerfield of electrophysiology, gets no respect, not taught in medical school or anything. So I'm from Dallas, Texas originally, and the football team, which just lost over the weekend, is the Dallas Cowboys. The basketball team is the Mavericks. In the spirit of being a cowboy or a Maverick, just because other people tried in the 1980s and failed, we're still going to give it a try. And so we basically found 3 key limitations. One was that this idea of placing the electrodes at the midpoint between the belly button and the bottom of the rib cage might have been a flawed piece of logic because the stomach, person to person, varies a lot. These are CT scans from human subjects. The 3 red dots are the suggested location from the 1980s. If you look at these 2 plots, you see those red dots don't cover the stomach. So there's a lot of anatomical variability. In addition, it turns out there can be a lot of artifacts whenever you place electrodes on the abdomen because of the musculature and the subtle subconscious movements that you make. So I won't bore you with the details of how we did the motion artifact rejection, but you guys saw Miro Medar give her talk, right? I did my training under her. We know how to estimate signals from artifacts and interference, so we can use principles from wireless and whatnot to do that, especially because the signal we care about is in a narrow frequency band. That really helps you. So you can develop procedures to do that using ideas from Wiener filtering. One thematic thing that actually occurred that's kind of related to about Muriel is, remember she talked about how they first estimated the noise and then they got the signal. Interestingly, the first thing we do is we estimate the artifact with Wiener filtering ideas, then we subtract out the artifact. So complete coincidence, but there's a theme there. Now, in order to capture the anatomical variability, well, you use a multi-electrode array, and you can also leverage imaging so you know where things are. So before we could play offense, we had to play defense, because remember, there are these papers that said doubtful clinical usefulness. So we basically took a clinical gold standard. If anyone has ever had to go to the doctor for GI problems after they do the endoscopy and everything seems normal, they send you over to nuclear medicine and they have you eat a radioactive egg sandwich, basically. And they basically do imaging on your abdomen to see how long it takes for food to empty the stomach. So what's unique about this approach is that the imaging does not expose you to radiation. It's just the egg sandwich. It's just an array of photodetectors on your abdomen. They typically capture stuff at 1 hour, 2 hour, and 4 hour mark, because a typical meal empties in about 4 hours. But we can capture images every second. There's no extra radiation. Got IRB approval to do this. We capture images every second. You can basically get a mechanical picture of the stomach contracting, which is this top plot. And we can compare that simultaneously with the electrical measure. And you can see concordance in up to like the third decimal point in terms of the dominant frequency underlying both of those. So this is just playing defense, saying that this is concordant with the clinical gold standard. Now, what we also got interested in is this whole idea. Remember, I told you guys that this electrical slow wave propagates, and ideally it propagates from top down. Well, let's see if there's variability in subject to subject and how that occurs. So we basically got a multi-electrode array, and after filtering the signal, what I'm showing you is just each color is a waveform coming from a different electrode. And one of the things that you see right away is that there's a phase shift compared to each of those. So if we take a look at the contours of equal phase, we can do all sorts of things using Hilbert transform to extract phase, and we can basically look at the direction that the wave is propagating. Something else that we can do is to, basically do things like controlling the false discovery rate to say, look, we have detected a wave. And if we remember from our electromagnetics class, those double E's of us where electromagnetics was required, I hear now it's not. If you have a plane wave, then the gradient of the phase, no matter where you are in space, should be always pointing the same direction. So basically what we can do is we can use Jensen's inequality and come up with a measure where you have the norm of an average divided by an average of norms. So it's always between 0 and 1. And the closer it is to 1, it means more of a coherent wave it is propagating. Control the false discovery rate, and then build a spatial histogram to see the direction the wave is propagating. So we did this not only in healthy controls, but also patients who had a diagnosis of GI problems. And one of the interesting things is we focus on patients where we had imaging, CT or MRI. And this patient, you see the end of the stomach and going towards the small intestines is about 170 degrees. We look at where those significant waves are, and lo and behold, they're primarily going in that direction. So it's like an anterograde propagating wave. In this patient with gastroparesis, here the small intestines is about 180 degrees. We look at significant waves, and what you see is this weird sort of bifurcation where there's almost no waves propagating towards the small intestines. Not surprising if you have nausea or reflux or something else, because the sequence of contractions is not propagating downward. So what we were able to show is that when we get this spatial information and about the percentage of time that there's abnormal slow waves, this correlated with the clinically indicated symptom questionnaire. On a scale of 1 to 10, how severe is your reflux? How severe is your chest discomfort, your upper abdominal pain, et cetera? And you can see this red line connecting to so many different things. So if we summarize this questionnaire, and they do this clinically until a single number between 0 and 5, this is on the vertical axis. So all that stuff about, so the more severe your symptoms, the closer it is to 5. And on the horizontal axis, we plotted the percentage of detected waves that are propagating in an abnormal fashion, not towards the small intestines. You notice in controls is that they don't have severe symptoms. The percentage of abnormalities is low, but in patients who have who are diagnosed with these problems, you see this correlation. So this actually was the first ever non-invasive measure of gastric function that correlated with the severity of symptoms. It seems pretty obvious to us, but if you think about doctors, what's the role of a doctor? Is to help you feel better. So if I want to come up with an objective measure, it should correlate with how you feel. So so one of the things we got interested in, this is where some fun math comes in, is we wanted to go beyond just simple, plane wave-like phenomena. What if there's more interesting types of wave patterns that are taking place? Can we start to look more at that? So at a very high level, at every electrode, we got the gradient of the phase. And we basically, so if you think about this, these are circular varrows because they go between 0, 360 degrees. And we can start to do things like building what these spatial histograms are. And we want to develop statistical models for this. So if we think about what is the canonical Gaussian-like distribution for something that's circular between 0 and 360 degrees, it's something called a von Mises distribution. So you can see here, this is a von Mises that has one mean. So the blue and green have the same mean. The green one has more variance, just like a Gaussian, the same mean and different variance. And likewise, the red and the orange have a different, same mean, different variance. This is what it looks like on a spatial histogram. And this is the equation of the density. This is sort of well-known, et cetera, et cetera. But in reality, our data could be more complicated, right? So what if our spatial histogram looks like this? All of us have heard of mixtures of Gaussians. You could do things like build mixtures of von Mises, right? So you can develop the similar types of formalities. Now, one of the things we got interested in is suppose that every subject we get data and they carry this probability distribution, this circular probability distribution, we want to be able to, compute distances between them. So we thought about computing something like a Wasserstein-like distance in the optimal transport. And what we're able to do in this framework is to, so people are familiar with this. This is the standard sort of framework. One of the key things we have to, so usually the cost in the Wasserstein distance is a notion of a distance on the basic metric space you're on, but keep in mind that 0 and 360 degrees are the same thing. So we don't want to use Euclidean distance. We want to use the distance on a manifold. Whoops, I don't know what just happened. And so at a high level, if the manifold is a circle, you basically look at the arc length in a counterclockwise direction versus a clockwise direction. Look at which one is shorter. And by doing that, we can compute these Wasserstein distances. And what we want to start to do is to develop things like clustering and whatnot between them. And if you remember, if you have points in Euclidean space, a natural way to build the center of a cluster is you take a look at all the points that are in the cluster and you just take the average, something simple like that. Now, I can use like a political analogy. Imagine that these are part of the same cluster and they're like voters and they carry a personality. Ideally, the politician that I elect should carry my personality. And I argue that if you take the average of the blue and the orange one, you get this red one. This does not carry the personality of each of the individual constituents. Each of these individual ones has one peak, but notice that the red one has multiple peaks. What if I instead could build something like the green one? Is there a way I could go about doing that? So the idea is that if we think about what the average is, we can interpret the average in a variational sense, that it is really just the minimum of the average distance between each of the individual points. You can see very clearly if you take the derivative, set it to 0, that's the variational interpretation of the average. Well, now if I have this Wasserstein distance between probability distributions, I can take the philosophical notion of the average coming from the variational interpretation. And when I do that, I indeed get this green curve. So basically, each of these are the spatial histograms of the directions from human subjects that are control subjects. And you see the peaks are slightly different. If I would have averaged them, we would have get something with many local maxima. When we use the Wasserstein Barrier Center, we get this blue one. So you see this kind of better represents its individual constituents in a political sense. So what's also nice about this procedure is it turns out you can solve with convex optimization. So this was the original picture about correlation with symptom severity. What we wanted to do was to really begin to understand that etiology, meaning what's underneath this. Is it the brakes? Is it the gas pedal? Is it other things? So what we were able to do was to use some of these features from this optimal transport stuff and clustering. And what we found is that in some of those subjects, their symptom severity exceeded a lot what was predicted from the electrical information that we got. If you think about that for a minute, that means that in these patients, it's suggestive that the cause of the car not working well is not the engine. Maybe it's the brake pedal, the gas pedal, something else. Coincidentally, in some of these patients, my colleague had treated them. And it turns out in these patients, he had treated them using treatments that, for instance, treat your depression, treat other things. And indeed, in those patients for which that occurred, their symptom got closer to the regression line. So this is just a suggestion that these approaches indeed can help us not only determine if the abnormality is in the stomach, but also outside of that as well. So this is a little fun thing I'm doing for Prashant. Part of my visit here is to make this to get different people in the audience interested. Sure, you had a question first. So here's an interesting thought, though. If you look at this patient over here, if you're on this part of the axis, our regression suggests your symptom severity is not that much. But see, your symptom severity is a lot more. sorry, so your slow waves basically are normal, but your symptoms are severe. My colleague, exactly. And then what was interesting is that you saw this occur in multiple patients, right? So is that helpful? So something else we got interested in, when you look at this data, we have this slow oscillating data. And one thing that's remarkable is that even in the diseased stomach, you always see these oscillations at about 0.05 hertz. It's just more that the spatial relationships between them are different. So imagine I take, every electrode, I look at it as an oscillating signal, I compute something like a Hilbert transform, and I see the phase, which is oscillating between 0 and 2 pi or negative pi and pi, however you like it. Well, what is the most canonical sort of idea of thinking about oscillations that are coupled? It's something for Sean to spend a lot of time in his career thinking about. It's the Kuramoto model. So think about why is the phase, so why dot is the rate of change of the phase. And you can imagine that is on average oscillating at an amount omega sub I, this is measured in radians. But the idea is that it's also kind of coupled to other oscillators. You could ask yourself, does this actually apply in the GI system? It turns out, yes. It turns out there's an interesting fun fact about these Cajal cells in our digestive system. They form a synctium, which means they share the same cytoplasm, and they also share the cytoplasm with your smooth muscle cells. So let's say they share variables, and so they're coupled. And so you could imagine envisioning a statistical model of coupled oscillators. So for people who don't know about the Kuramoto model, this is like the canonical example of nonlinear dynamics from which you can actually analyze things. So the analogy that people like to give from a control perspective is imagine oscillator J and oscillator I were the exact same phase at time t minus delta. Well, if you stand back and you look at this model, then what this means is that because this is signed right here, that means that there's no influence from the other oscillator because theta J minus theta I is 0. So he just continues to oscillate at omega sub I, oscillator I does. Now imagine at time t, oscillator J is now a little bit ahead of oscillator I. Then if you look at this right here, if you remember this, sine U is approximately U when U is near 0. So the sign of this difference is approximately the difference. Let's assume for now that this coupling kappa is greater than 0. Then what this is saying is that if I'm oscillator I, oscillator J is a little bit ahead of me, then my rate of change should be the intrinsic rate of change, but go a little bit faster because he's ahead of you. So you can start to see how this can allow for these coupled oscillators to be related to one another. This is well-known types of stuff. So this is the original model of oscillators, but we know our measurements are intrinsically noisy. There's other stochastic phenomena. I wanted to come up with a stochastic model and from the data to try to infer the coefficients. So I thought about basically a stochastic differential equation where we have everything the same, but you put a little bit of your standard Brownian motion there. Now, one thing we have to remember is that the left-hand side here, so if I just unravel the derivative, right? Notice that the left-hand side is phase between 0 and 2 pi. So when I take this Brownian motion, I have to put a modulo 0, 2 pi here. Now, when I do this, if I sample in an amount delta, which is very small, then it turns out that a wrapped Gaussian is basically a von Mises distribution. And you can show this, that they converge in a very rigorous sense. So I can interpret this additive amount as that von Mises that I showed you before. So what that means is the conditional distribution of the oscillator at time t plus delta, given the path of all the oscillators, is just a von Mises distribution centered at the deterministic dynamics, and it has a variance that's proportional to 1 over delta. This is like using standard, stochastic differential equations, blah, 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 blah. So suppose I'm going to look at the probability of all the data, right? Well, according to this model, you can write down and use the chain rule of conditional probability. There's appropriate conditional independencies. If I focus on the ith oscillator conditioned upon all the paths, it turns out after I do some trigonometry, it can be exactly described as a term that looks like this, where there's a term u sub I, and there's a sine and a cosine. Now, what's interesting is that if I unravel these terms, remarkably, this is the equation for what u is, okay? And notice that there's a delta in front of it. And delta is just basically the time between two successive samples. So what's interesting is suppose I end up sampling at a rate much faster than the rate of the intrinsic oscillations, then I can just use second order approximations to sine because notice there's a delta in front. So that means that these terms are near 0. So it's sine of u in both of these cases and cosine u. Sine u when u is near 0 is approximately u to second order. Cosine is 1 minus 1/2 u squared, which means that when I plug all this stuff in, this is now quadratic in kappas. The kappas are what I want to infer from the data. So if I actually want to do maximum likelihood estimation,
as long as I sample sufficiently fast. This is a quadratic estimation problem with a closed form solution, linear, just matrix inverse. So it's a theorem. You can say that rigorously. What's also nice is that you can do all sorts of extensions. We can actually get a closed form expression for the Fisher information, for the variability in our estimates. But we can also do things by looking at what if the dynamics, what if the coupling actually changes dynamically? We can implement, nonlinear filtering algorithms for that, provided that you sample sufficiently fast. So I find this remarkable, but as much as we understand, this has not been found before. So as much as you think about the Kuramoto model is nonlinear, the inference problem, provided you sufficiently sample, is actually linear in a very rigorous sense, right? Because there's uniform convergence, right? So we applied this to some data, and you see very interesting findings. So now what we're estimating is a matrix, where kappa ij is basically the coupling between spatial location I and spatial location j. And when you take a look at healthy subjects versus unhealthy subjects, you see these interesting structures. And what's also cool about this is if you think about what underlies the wave equation, the structure of this matrix, you can unravel from that. So this is just a little fun thing for Prashant. So, the last sort of limitation that we tried to surpass is dynamics are not captured. Imagine you waited 6 to 9 months. Prashant, figured you'd have a question. Yeah. Oh, so right now, imagine that this is the data that we record from our multi-electrode array, and every electrode generates a waveform. So we always filter to around 0.05 hertz, because we've done studies on over 100 subjects. It's consistently right around 0.05. So we find that the intrinsic frequency, we filter within there, and then we do a standard Hilbert transform to extract the phase. And if you look at the phase across different electrodes, you see these phase offsets, right? These differences. So we want to capture what the nature of that is, and we do that according to this statistical model, right? So it's another way to kind of get at the underlying inherent spatial dynamics of what's occurring. So the last thing is that, so we started off building, we actually just use a conventional off-the-shelf EEG amplifier. These signals are at the level of microphotons, about a factor of 10 larger than EEG. And they're very low frequency, 0.05 hertz. So as long as your EEG amplifier is DC coupled, you can literally acquire this data from an EEG amplifier system. Sheree, yes.
Subjects, the gold standard contacts, right? The 3
Yes.
Everybody's CT scan something.
So
So
that's actually what we did. So in that paper I showed you with the correlation, and all those patients, we had CT or MR on all of them, and we placed the center of our array on the antrum of the stomach. Now, moving forward, thanks to technology, you can now use a portable ultrasound device that only costs $2,000. You can plug into an iPad or an iPhone, called the Butterfly Network. Any board certified doctor can get it. So a bunch of my clinical colleagues bought one, and you can find the antrum of the stomach from a portable little wand device that you'll see on the slide. Yeah, so you can get the location of the stomach easy, thanks to technology now. So you don't even need an MR or a CT. So we wanted to go beyond just capturing data in the laboratory environment, because sometimes your symptoms are dynamic. Imagine you waited 6 to 9 months to see the GI specialist, and when you show up, he runs a test on you, but you don't have symptoms that day. That would really suck. So the idea is, well, what if you could take a device home with you and track your symptoms? So we totally took advantage of, it was more appropriate when I was at UC San Diego. We rode our surfboard over other people's wave of innovation. So when the Brain Initiative came out, Texas Instruments built this amazing little chip, the ADS 1299. Some people are aware of it. It's basically the same thing as a $30,000 EEG amplifier in a small little chip, because they envisioned with the Brain Initiative, there's going to be so much innovation with portable EEG. You can just rig that thing a little bit with resistors and capacitors to make sure it doesn't attenuate low frequencies. And you can MacGyver the system, and you have a system for measuring the electrophysiology of the gut. So we did that, and we basically built a smartphone app to showcase, so you could determine when you had a bowel movement, a meal, a symptom, et cetera, artifact rejection, spectral analyses, all sorts of fun. So again, I'm really talking to the audience here. So this is a new finding, and the first author on this finding is someone that did her undergraduate studies under Sri, named Sandhya Subramanian. She stayed in the family for a long period of time. She had her undergrad studies was free, her PhD at MIT and her Emory Brown. And then Emory told me, Todd, you should get your hands on her because she's coming out west. Her husband lives in the Bay Area. And so she's a postdoc with me at Stanford. So something that she was able to do is this data that we collected with this off-the-shelf device. The device has a gyroscope on it as well. And we took advantage of two things. Whenever you place electrodes on the abdomen, the first thing you do before you filter, you see the heartbeats. You see the R intervals of the EKG. So one thing that's interesting is, remember I told you about the gas pedal and the brake pedal, and that's autonomic nervous system. The stomach is modulated by the autonomic nervous system, but so is the heart. So can we actually look at the heartbeats to get a window into what's going on with the autonomic nervous system to see in what situation is the cause of their problems due to the brake pedal and the gas pedal? And specifically, you want to identify windows of time where you know that the heart's engagement by the sympathetic nervous system versus parasympathetic differ. One of the things we know very well, if you talk to a cardiologist, they'll say be very careful about the heart because the heart is always responding to a whole bunch of different demands. So if you're just monitoring the heart when someone's awake and living, you got to be careful. But during sleep, it's well known that you get into more of a parasympathetic state as compared to awake. So the first thing we have to do is to figure out a way just from this device on the abdomen to determine when someone is asleep or awake. And Sandy saw that problem. She presented it at the big biomedical engineering conference last year. I won't bore you with the details of it, but here's a healthy human control. Here's someone with diabetic gastroparesis, where it's more dark means where you're asleep, otherwise you are awake. And some of you people who are familiar with heart rate variability, one of the things we know is that if you look at the heartbeat dynamics, and we actually use a point process formulation that was developed by Emory Brown's group. It's more rigorous. You can get millisecond by millisecond measures of what's going on with the heart. At a very high level, this middle plot that's called the sympathovagal, when it's high, it means it's more sympathetic, and when it's low, it's more parasympathetic. Notice when this healthy subject goes to sleep, you see this huge drop off, which means the denominator got bigger. There's more parasympathetic innervation. This is bread and butter, what we know physiology. When you're asleep, there should be more parasympathetic innervation. If this patient who has diabetic gastroparesis, you see almost no change in awake versus sleep. So sort of the regulation of the autonomic nervous system is really off in this patient with diabetic gastroparesis. So we were basically able to get four features: the intensity as well as the regulation, where here we look at sleep versus awake. And from our GI measures, we looked at meals, because after a meal, your stomach should really kick on, right? Versus when you were not. So we have these, sleep versus wake, before a meal, after a meal, and we get intensity and regulation. And by combining these features, we can basically separate subjects with gastroparesis due to diabetes without, et cetera, and healthy human subjects. And we can separate healthy controls versus patients, 17 of 19 of them, and diabetic gastroparesis versus non-gastroparesis, 9 out of 10. Slaughter point is interesting because determine whether or not someone has diabetes easily, you just do a little blood test, right? Half of diabetics go on to have gastroparesis and half don't. Imagine if early on you could basically predict whether or not that's going to occur. That's why that bottom point is interesting. So this is under review. So the last thing that we did really fast is, I've done some work on building flexible electronic technologies. I won't bore you guys with the details, but we were actually able to build flexible electronics. So imagine you almost have like a Band-Aid you can place on the abdomen. You can measure this information. We were able to do that. Sure, going back to the imaging, this is us using this butterfly network device. It costs $2,000. You plug into an iPad, you can find the antrum in the stomach, and you place the electrodes there. Now, this is a message kind of sort of to Muriel and Sean, because I remember when I was younger, Muriel actually alluded to this in her talk, this whole idea about optimizing the constellation for signaling and whatnot. There's an interesting question that comes here. The second that you can build an electrode array using microfabrication technologies, imagine that I have the same spatial area, like the area of my phone. I could either put a small number of big electrodes to fill up this area, or I could have a large number of very small area electrodes. And I could have so many different combinations. What is the optimal combination, and can I do it in a subject-specific manner? So we went about trying to attack this problem, and the first thing we wanted to do was to understand how does the property of the electrode change with its area? And so you can do something called electrical impedance spectroscopy, where basically you shove in voltage, you measure current, you can look at what happens, you look at the, across different frequencies. And so you can look at the complex from a complex perspective. Something that's well known about the electrode skin interface, this is like bread and butter electrophysiology called the Randles circuit model. And there's one property here, which is a capacitor. And if you think about a capacitor, there's the area, right? If you guys remember the formula for a capacitor, right? It's epsilon naught area over H. Well, if you have a wide electrode versus a small electrode, the areas are different. So what we can do is basically do estimation using this procedure for our different types of electrode chemistries. And we can figure out, so I can write this as some constant times the diameter of the sensor squared, right? Because the area of a circle, right, is proportional to the diameter squared. I can estimate this constant, and then I can predict how, if I change the diameter of the electrode, how the impedance will change. And we can confirm this with data. So the red points are what we measured, and this blue is what we estimated. You can see our estimate is consistent with that. Now, what's cool is you can vary across arbitrary diameters and see how the impedance will change. Now, if I know how the impedance changes at arbitrary diameters, I can do all sorts of different things. And I won't bore you with the details, but what's interesting is if I can pinpoint the location of the slow wave abnormality, there are actually pacing technologies and other things that can help you. So, you can imagine there are all sorts of therapies that can help if I can pinpoint the location of the problem. So I can basically turn this into a source localization problem, solving an inverse problem. But the key thing I need to figure out is if I use an array like this versus an array like this, I always have that signal noise term. That signal noise term is intimately related to the impedance, right? From the basic thermodynamics, if people remember Johnston noise and the thermodynamics of the noise related to the impedance. So I won't bore you with the details of this. We developed some inverse estimation procedures, but the key thing that I want to do was interesting was to separate, do I want a small number of fat electrodes or a large number of skinny electrodes? And we used some Fisher information optimization procedures, and just like what we hypothesized would occur, did, which is really cool. So for every model across different electrode diameter, we actually using our electrical impedance spectroscopy estimation results. So we know how the impedance varies. If you know how the impedance varies, that is in one-to-one correspondence with your noise variance. So now if I have different noise variances, I can look at basically the Fisher information and trying to estimate what's inside the stomach based upon the recordings. We hypothesized, and indeed this comes up in the data, that so these are two different human subjects with different CT scans, and we can see the maximum of the Fisher information, where basically the minimum variance unbiased estimator is the best. And when you're in a situation where the electrode diameter is low, that means that you have really high spatial resolution, but guess what? You're gonna have a ton of noise, because it turns out that it's sort of well known that when you have small area electrodes, the measurement noise is larger. And on the flip side, if you have very large diameter electrodes, you're in this regime. So fine, your noise variance went down, but you don't really have any spatial resolution. You cannot resolve anything spatially. So what's cool about this is I call this like bring your own anatomy and bring your own electrode chemistry, because our methodology applies to any electrode chemistry. We can do this and we can figure out the optimal pattern. So this is part of this is I want to present this because it is in the spirit of Muriel and Sean and some of their signal constellation optimization stuff. The details are different, but that's the the high level story. So the last thing I'll talk about very briefly is I primarily talked about the gut, but remember I originally started off studying the brain with Emory Brown. We all know the gut and the brain are connected. Can we do fun things with both of them? So we're in this new building, at Stanford. When I moved there about a year and a half ago, I should be doing new research. So we completely stumbled upon this, but there's interesting group out of France, interestingly, Muriel. And remember at the ISIT in 2019, I snuck away to visit this French researcher, this lady named Catherine Talon-Baudry, and her research group had this fundamental discovery. This was the first ever discovery that there is an electrical coupling between the brain and the stomach. And so what they did is they used an MEG scanner, which is like an analogy to EEG, but it's magnetic. It has higher spatial resolution. And they used the same type of methodology that we use on the stomach, the electrogastrogram. And what they found was that the phase of the slow signal modulates the amplitude of EEG, specifically in a frequency band called the alpha band. Fascinating stuff, right? So the stomach and the brain are coupled. They did this in the resting state. That's why I mentioned the point about the default mode network, Sri. Yeah. So the stomach and the brain are coupled while you're at rest in this phase amplitude coupling sense. Now, that's the good news. Here's the bad news. Here's the location of where there are MEG scanners in America. And notice we're giving a talk here in Florida. All the states that are great means there's not a single MEG scanner in the whole state. So if we want this science to really be broadly adopted, we cannot depend upon MEG, right? For instance, California has two. There's not even one at Stanford, right? You know, Stanford has this big multi-billion-dollar endowment. There's one at UC San Diego, UCSF. Stanford doesn't even have one. So what I got interested in doing is saying, well, let's replace the MEG scanner. Can we basically just use EEG? Before I move on, I believe there was a question. Okay. Absolutely, yeah. So MEG is clinically useful for many things, for diagnosis of, you know, PTSD, for many things.
Goes down, you're tired. So I now understand why, of course, the brain, when you eat, you get tired.
They call it the itis. Really, many things they call it, but yeah.
Well, it's consistent.
Yeah, and then that's why I kind of made those comments after Shree's talk about the whole performance and the internal states. Some of this is actually driven by visceral signals. So Catherine Talon-Baudry, who's one of my scientific heroes, the lady I visited when I was in Paris at the conference, she has written just all these papers about how visceral states shape cognition. And building upon what Shree said, even within neuroscience and motor neuroscience, they didn't pay attention to other brain areas. And analogously, in cognitive neuroscience, they would only pay attention to the brain. But it turns out that in the spirit of Sean's workshop, we know from control that the second that you share variables, things are coupled. And so we know that the brain and the gut, the brain and these different systems are connected. And in some situations, they play an important role in shaping what's going on in the brain. So yeah, her work, she's been publishing all these papers on it. Fascinating. So here's her work from 2017, and this is where there's phase amplitude coupling. The more red there is, there's more. We published a paper with EEG, showing we can kind of sort of pick up analogous information just in December. And then just earlier this week, we did a study with a graduate student. You can't see carefully, but she's wearing a brass rat. She's a first year Stanford grad student, undergrad ECS MIT, and you can see similar brain regions here, sort of this right upper area, and then this sort of bottom parietal occipital area. So this is very exciting. That and by the way, this was 32 channel EEG. This is 128 channel EEG. So this is in the new building that we're at Stanford. They have an opportunity where anyone on campus can get access to these resources. So for the purpose of time, I'm going to move fast, but how do you quantify phase amplitude coupling? There's a paper on this by Nancy Capel's group at Boston University. They developed an approach to do this, but if you really think about it from a rigorous statistical sense, it doesn't completely pass muster, in my humble opinion, like Emory Brown would not be completely smiling. So this Todd et al. Paper has over 1,000 citations. It's kind of like the gold standard for measuring phase amplitude coupling. We wanted to really come up with a statistical measure that you can test goodness of fit and whatnot. And it turns out you borrow ideas from Norbert Wiener. So Norbert Wiener, as we know, cybernetics, and he was hanging out with people at Harvard Medical School. So he actually measured his own, not only brain waves, but alpha waves, it turns out. So if you look at some of his old papers, he developed, state space models for modeling EEG and whatnot. And here's like a simple example of this. So if you take a look at the spirit of this, this is sort of a Gaussian state space model. If you think about the amplitude of that, if we remember from first glances, if you take the amplitude squared of the sum of independent Gaussians, you get something that's like a gamma distribution, right? So we thought about modeling the amplitude of the EEG in terms of a gamma distribution, amplitude squared. Now keep in mind, what's a natural measure of coupling between things? Well, a mutual information. It's not negative, it's zero if and only if the two are statistically independent. So the idea that we came up with was we have X, which is the phase of the gastric signal, and then the amplitude of Y, right? So I put the absolute value of Y, but keep in mind, we care about the amplitude, which is non-negative. That the square are invertible functions of each other. So the mutual information is invariant with respect to invertible transformations. So we can play around with the amplitude squared. It's more convenient. And then again, using this idea from Norbert Wiener, if you're taking sums of squares of Gaussians, you get stuff that's gamma-like. What if we build a generalized linear model with a gamma-like approach? And I won't bore you to the details of this, but if we structure it in a certain way, we can make the model fitting procedure convex and all of that. And we can actually do statistical goodness of fit to really see, does the joint distribution match the data? And it does. Then we compute the mutual information. And the takeaway lesson is that when we simulate synthetic data, our procedure outperforms the Todd et al. Paper that has over 1,000 citations. So by kind of like the takeaway lesson is, you know, really think about a model that reflects that's consistent with the data, compute something like the mutual information as a measure, and it actually works even better than something with over 1,000 citations. So this was just presented by my student Andrew over the summer. Now, if there is phase amplitude coupling, what does this mean? What's underneath this? You know, neuroscientifically, you want to ask this question. So, okay, we know the stomach and the brain are connected. What does the literature tell us? Well, we know anatomically, and here's a review paper by a guy named Terry Powley at Purdue. We know that goes from the enteric nervous system through brain stem areas, for instance, via the vagus nerve. And he talks about the key cortical area involved is the insular cortex. Then there was another beautiful paper in 2020 in PNAS where they just discovered anatomically what cortical areas send descending projections from the brain down to the stomach. And one sentence in the paper is the anterior insula is the major cortical source. Interestingly, that phase amplitude coupling, where there was lots of coupling, guess what it overlapped with? The insula. So the insula appears to be a region of interest. So what is interesting about the insula? So here's a February 2007 New York Times article, small part of the brain, profound effects. You know, you resect part of the insula, people give up cigarettes, long neglected brain region, social emotions, lust and disgust, etc. So it's tied to social functioning. What's also interesting about the insula, and I know this because I had a postdoc from Chile way back in like 2008 when I was at Illinois who studied a topic called interoception. So, you know, there's an old Greek adage that says, in order to know others, you must know thyself. So the insula we know is implicated in social emotion stuff. It turns out it is the main brain area that receives internal inputs from visceral organs. So interoception is a multidimensional construct that reflects the perception of the internal state of the body, such as hunger or satiety. So it turns out that interoception is this huge, interesting topic now. NIH has special calls on it, yada, yada, yada. It's implicated in drug addiction, alcoholism, anxiety, obesity. That's not surprising. You never feel that you're full. Eating disorders, you never feel that you're hungry, right? So interoception is a really hot topic. And the main clinical procedure that they use to assess this in eating disorder clinics is completely behavioral. So what they do is they have you drink some water and they say, drink until you feel satiated. Then they say, I want you to drink more water until you feel full. The idea is that if you have good interoception, satiated and full should be the same thing, which means the extra amount for full should be small. So you create this ratio, and if you have good interoception, this extra amount is small, the ratio is near 1. So this is a clinically indicated measure of interoception that's used in the eating disorder clinics. I'm interested in physiologic measures. So over the summer, I had an awesome undergraduate student work with us, and we basically wanted to see if this measure of phase amplitude coupling, which you can get with EEG now, which means this could be done, this could really be done globally across the world. It could be done in the state of Florida, it could be done, you know, et cetera. And this is very preliminary work, but so far for human subjects, what we see is the behavioral measure of interoception correlates with the increase in phase amplitude coupling, you know, post drinking water as compared to before. So this is ongoing work that we're exploring, but the idea is to come up with physiologic measures of gastric interoception, which could help with eating disorders, with obesity, many different things. And so this is sort of full circle. So as I conclude, one thing that sort of underlies how I think about our research topics is call it red ocean, blue ocean. So red ocean is our areas of research that are very saturated, people using the same techniques. I don't like to play in those areas. If I'm going to go into a saturated area, I either like to come in with a new personality, like what Muriel did with Ken, coming in with, you know, I mean, they attacked this coding problem. They didn't use the same techniques as everyone else. They came in with a new perspective, right? Or you find a completely new area that no one is thinking about, as long as you convince yourself that it's important. And that's kind of what the blue ocean represents. So that kind of underlies our philosophy. So with that, I conclude.